Welcome. In the last video, we understood the role of abiotic and biotic agents in pollination. Now, in this video, we shall try to understand what are outbreeding devices, and also we shall see about pollen pistil interactions. So now, let's see about outbreeding devices. Now, what are these outbreeding devices? So let's define this first. So these are various mechanisms developed by the plants to avoid self-pollination. So these are various mechanisms developed by the plants to avoid self-pollination. So I hope we remember that what actually is this self-pollination, that the transfer of pollen grains from the anther to the stigma of the same flower. So in that case, the condition we call it self-pollination, right? Now this will take place, of course, within the flower present on the same plant. Now this self-pollination does not yield much variations. So it does not produce much variations. I think you still remember in the last chapter that what we have studied, that variations are very much important for the survival of the offspring. Now in case of this self-pollination, right, the variations are not produced much, right? And for this reason, most of the plants would like to go for cross pollination. So in case of cross pollination, it results in the production of variation. So it produces, right, variations which are very important for the survival of the offspring. So self pollination will not produce much variations, whereas cross pollination produces variation. So that is the reason why most of the plants like to go for the cross pollination. Of course, not only the plants, other organisms also like to go for cross fertilization instead of what I say, the self fertilization, right? Because the self fertilization will not produce much variations, and in case of only the cross fertilization results in production of variations which are very important for the survival of the organisms, right? So now we understood that the plants, majority of the plants do not like to go for self pollination, and they will avoid this self pollination, right, and like to go for only the cross pollination. Now in case of the plants that have bisexual flowers, and there are unisexual flowers present on the same plant, so in this case they like to avoid this self pollination. So if there are bisexual flowers on the plant, then if that anther, I mean what I say, the male reproductive organs and the female reproductive organs, if they mature at the same time, then there is a chance of self pollination. So they like to avoid this self pollination, and they develop certain mechanisms which I am going to discuss shortly, right? Now let's see what are all those outbreeding devices developed by the plants to avoid this self pollination. So the first one is the pollen release and stigma receptivity, right, is not synchronized. So pollen release and stigma receptivity is not synchronized. It means here synchronization means taking place at the same time. The male reproductive organ and the female reproductive organ, if they mature at the same time, then I say they are in synchronization. And if they don't take place at the same time, say the male reproductive organ matures, okay, in one time, and after that the female reproductive organ may mature, or the vice versa can take place. Means in this case both the reproductive organs do not come to maturity at the same time. So then I use the word not synchronized, right? So pollen release and stigma receptivity is not synchronized. That is one point which I will be discussing in detail shortly. And another mechanism is that the anther and stigma are placed at different heights. So anther and stigma are placed at different heights, or I can say positions. So this also I will be discussing shortly. Just I am listing out the various outreading devices. So the first one, which I said already, the pollen release and stigma receptivity is not synchronized. The second one, anther and stigma are placed at different heights or positions. Then third one is self incompatibility. So self incompatibility, right? And the fourth one is the production of unisexual flowers. Production of unisexual flowers. So, right, these are the outbreeding devices which I'm going to discuss. So all these, will help in preventing the self-pollination. So if the pollen release and stigma receptivity do not take place at the same time, then there is no chance of self-pollination. And if the anther and stigma are placed at different positions, then also there is no chance of self-pollination. And all of these will favor the cross-pollination, even self-incompatibility and the production of unisexual flowers. So all these mechanisms, right, favor the cross-pollination. So I can say that all these prevent self-pollination, and favor cross-pollination. So they are going to prevent the self-pollination and favor the cross-pollination. Now let's discuss each of these outbreeding device a little detail. So first one, let's say about the pollen release and stigma receptivity not synchronized. As I already mentioned, that not synchronized means not taking place at the same timing, right? So now here when stamens and carpels mature at different times, so the stamens may mature earlier than the carpels, or carpels may mature earlier than the stamen. In both these conditions, what happens is it will avoid self-pollination. So point is that in a bisexual flower, so this is the stigma, and say here is the anther, and say that if both of them mature at the same time, then there is a chance of this pollen grain coming and settling on the stigma of that flower. So then it can result in the self-pollination, right? So now if the plants have developed a mechanism that the stamen, so we know this is the stamen and this is the pistil, right? So if this stamen matures right before this pistil or the carpel, then right, even though the point is that the pollen grains they fall on the pistil, there will be no chance of fertilization, right? Or vice versa can also, what you say. But in this case, if this pistil does not mature but stamens have matured and the pollens have released and fallen on the stigma of that pistil, then there is no chance of the pollen germination on this stigma. So hence in this case, this will completely avoid the process of self-pollination. So that's what we say, when stamens and carpels mature at different times, right? Then of course such a condition we can use a word dichogamy, right? And this, of course, the dichogamy is a word which is not mentioned in the textbook, right? So here this dichogamy can be of two types. One I call it protandrous condition, and another one called the protogynous condition. So I'm just giving a little more explanation for this statement that pollen release and stigma receptivity not synchronized, right? Now this protandrous condition, I think in the last video I have given these words, protandrous and protogynous. Now in this protandrous, which means the stamens, means the male reproductive organs mature earlier than the female. So stamens mature earlier than carpels. So in that case, the condition is called protandrous. So we can see this protandrous in case of plants like abelmoschus, then helianthus, then coriandrum, etc. are some examples in which we see even though they have these bisexual flowers, they show the protandrous condition. So where you will see that stamens are going to mature earlier than the carpels. Now coming to the protogynous, which is opposite to that. So where we see in this case the carpels mature. So here protogynous means the female, androus means the male. So the female reproductive organs will be maturing earlier than the male reproductive organs. The condition becomes protogynous. So carpels mature earlier than stamens. So if the carpels mature earlier than the stamens, the condition is protogynous. So we can see in case of Michaelia, right, Annona. So in case of the plant Annona, right, and then we can also see in case of Mirabilis, etc. are some plants in which you will find this protogynous condition. So all these, that is either protandrous or protogynous, or simply I can say the dichogamous condition, will prevent the self-pollination and favors what I say the cross-pollination. So as I already mentioned, that pollen release, sigma receptivity, when they are not synchronized, obviously it will prevent the self-pollination and will favor, okay, the cross-pollination, right? So this is the first point, the pollen release and stigma receptivity not synchronized. So the next one, the next outbreeding device, let's see that the anther and stigma are placed at different positions. So the second outbreeding device is the one that prevents the self-pollination, okay, is anther and stigma are placed at different positions. So it means that the stamens may be, what I say, having a longer filaments, right? So that keep the anthers well above the stigma, or the vice versa can take place. The filaments may be much shorter and placed below the level of stigma. So both these conditions will help in preventing the process of self-pollination, right? So now let's understand this anther and stigma placed at different positions by an example. So let's see a condition called heterostyle. So heterostyle means the length of the style is different. So this heterostyle is seen, okay, in a plant called, okay, Primula vulgaris. So it is seen in case of a plant called the Primula vulgaris. So where you find the length of the styles will be different. So this Primula vulgaris, right, it's commonly called the primrose plant, and this produces dimorphic flowers. So it produces dimorphic flowers. So heterostyle, so where it simply indicates that different lengths of style is seen. So it produces, as I said, in case of this Primula vulgaris, it produces two types of flowers. So where you see one type of flowers will have long styles, okay, so it produces long styled flowers, and another one with short style, so short styled flowers. So long styled flowers and short styled flowers. So now if we see in this Primula vulgaris flower, so let's see. So here we have the ovary, and this is the style, okay, and you can see the stigma present at the top. So this is the stigma. And now when we see the stamens, okay, so you can see in this case the stamens have a very short, filament, right? So these are stamens, and you can see this is the position of the anther. So it is placed much or well below the stigma, so that what happens is the pollen easily doesn't get settled on the stigma, so thereby avoiding the process of what I say self pollination, right? So in this case, so this one is the long style. So you see in this case the long style. Now let's see in another case, so where you find the style is short. So right, you can see in this case, so this one is the stigma, of course this is the style, so that is a short style, is short style, and this one is anther, and this is its filament, right? So in this case you can see that anthers are placed well above the stigma. So in both these conditions you can see due to the difference in the length of the style puts that stigma at different heights, and this will prevent what I say the self pollination and favors the cross pollination. So that's how we see the dimorphic flowers in case of Primula vulgaris. Right, now let's see third outbreeding device. The third outbreeding device is self incompatibility. Right, now what happened in certain plants, they developed genetic mechanisms to recognize whether the pollen has come from the same flower or from another flower present on the same plant. So if it is the case, if it is able to identify the pollen coming from from the same flower or from another flower of the same plant, then they will reject and thereby avoiding the self-pollination. So I say that the self-incompatibility, which means plants develop certain genetic mechanisms to identify the pollen, right, whether it has come from same flower or another flower present on same plant. So in that case, so they are going to identify that what I say the pollen, and it will not allow the pollen to germinate or to grow the pollen tube, okay, in its pistil. So that's what we call it self-incompatibility. So let's see here how this one takes place. So here we have a plant. Okay, so you can see so this is the pistil, and you can see here the anthers, and here is another flower. Okay, right. So now in this case, so let's see. So as I mentioned that in case of self-incompatibility, right, if the pollen grains from this anther come and settle on this stigma of the same flower, so in this case the plant will immediately recognize, okay, because of it has certain genetic mechanisms that has a ability to recognize whether the pollen has come from the same flower or from another flower present on the same plant. So in this case, this pollen may also come and settle on this, right? So in both these cases, it's going to identify that pollen and then reject that pollen, right? So if pollen comes from same flower or another flower, so another flower of same plant, then the pollen germination or the pollen tube growth in the pistil is prevented. So it stops the pollen germination, or if that pollen germination already happened and then pollen tube started to grow, but even then it will not allow the pollen tube to reach the ovules of that flower. So that's what I say, if it comes from the same flower or from another flower of the same plant, then the pollen germination is stopped, or if already the pollen germination has occurred, then the pollen growth will be stopped in the pistil. So this is what we call it self-incompatibility, and the self-incompatibility is another outbreeding device which helps in preventing the self-pollination and favors only the cross-pollination. Right, so let me give a few examples in this case. So we can see this happening in case of solanum, in case of passiflora, etc. in such plants. Okay, so they have developed genetical mechanisms. With that, they are able to identify the pollen whether it has come from, okay, same source, okay, and then preventing the process of self pollination. Right, now let's move on to the last outbreeding device, so that is production of unisexual flowers. So the last outbreeding device is the production of unisexual flowers. So production of unisexual flowers will also help in preventing the self-pollination and favors the cross-pollination. Right. So in this case, the flowers are either staminate. So they are either staminate or they are pistillate. So they are either staminate or pistillate. So here staminate means we know they have only stamens, and in this case this is a male flower, and in this case the pistillates, pistillate flowers, which mean they have only the pistils, and in this case these are called the female flowers. So now these male and female flowers can be developed on the same plant, or they will be developing on, okay, different plants. Right. So uh, right, so let's see, here we have a plant, so that has both okay, staminate and pistillate flowers. So here say this is one that has right, so only pistils are present. So what I say, in this case this is a pistillate flower, also called of course the female, the flower. Right. Right, so this is the pistil. Right, now let's see, so this is another flower, and has presence of the stamens. Right, so these are stamens, and in this case this is a staminate flower, so that is a male flower. Now a plant that has both pistillate as well as the staminate flowers is actually a monoecious plant. So I say a plant that has both male and female flowers, right, then we have to call that plant as a monoecious plant. So it's a monoecious plant, or I can say it is a bisexual plant. So monoecious plant or the bisexual plant here. Okay, right. So in this case you can see the presence of unisexual flowers on the right same plant. Now in another case you may find a plant may have only the male flowers. So the staminate flowers are present on one plant. So you can see in this case, so this is a right male flower, and of course here also you will see the presence of right stamens. Right, so I can say this one is a male plant. So this is a male plant, and similarly we may also have a female plant, so that will have what I say the pistil. So in this case, right, so this is what I say the female plant. So this is the pistil present. and these are stamen. So you can see that there are male means any flower on this male plant will have only the staminate flowers, and any flower on this female plant will be only the pistillate flowers, right? So that's why I use the word, okay, the male plant and female plant. So in this case the plants are actually, right, dioecious, or I say these plants are unisexual plants. Okay, so dioecious or unisexual plants. Right, now let's see, coming to the plant that has unisexual flowers. So in this plant, what we see, right, so in this monoecious plant, right, the presence of pistillate and staminate flowers on the same plant. So in this case it prevents autogamy. Of course autogamy will not take place, but it will not prevent gytenogamy. It prevents autogamy but not gytenogamy. So I hope we remember that gytenogamy is the transfer of pollen from anther to the stigma, of course from one flower to the stigma of another flower present on the same plant, right? So that's what we call the gytenogamy. So in the plants that have unisexual flowers, so in that case it will prevent the autogamy, but it will not be able to prevent the gytenogamy, right? Now coming to this unisexual plants. So this is a, okay, and just write here. So this is a unisexual plant or a dioecious plant. So in this case the unisexual plant or dioecious plant where the male and female plants are completely separate. So in this case it prevents both autogamy and gytenogamy. So it prevents both autogamy and gytenogamy and favors, so what I say, the xenogamy. So in this case what occurs is the xenogamy. So now we understood that the last outbreeding device, which is a production of unisexual flowers. So this production of unisexual flowers will also help in preventing the self-pollination and will favor the process of right, cross-pollination. So this is all about the outbreeding devices, right? So now let's see the next concept, that is pollen pistil interaction. So the next concept is, so pollen pistil interaction. So how the pollen grains, okay, interact with the pistil. So let's see that one. So here the pollen pistil interaction, so we can define that the events, so whatever all the events from the pollen deposition on the stigma, so all the events from pollen deposition on stigma until, okay, the pollen tube. So once the pollen deposits on the stigma, it gets germinated and results in the production of a pollen tube, and that pollen tube enters the ovule. So whatever that events that take place, okay, from that pollen deposition on the stigma and the production of that pollen tube and growth of that pollen tube through the stigma and the style, and finally entering into the ovary and reaching the ovule. So all that events will come under this pollen pistil interaction. So I can define this pollen pistil interaction as all the events from pollen deposition on the stigma until the pollen tube enters the ovule. So now let's see. So point. Let's see the explanation in this case. So here, this is the stigma here. So during the process of pollination, there is no rule that the pollen grains of the same species, okay, have to come and settle on the stigma. So when we see on stigma, right, so this stigma may receive the pollen grains from, of course, from the same species, or it may receive from the same flower, it may receive from, say, another flower on the same plant, or may receive from another, okay, flower on another plant but belonging to the same species, or it may even receive the pollen from, okay, another species, right. Now point is that in simple what I say here, it will receive the pollen grains, okay, from various sources. So here I am just representing with various colors that indicates these are the pollen grains from various sources. So these are the pollen grains from various sources that settle on the stigma. So I say that the pollination does not guarantee the right pollen to come and settle on the stigma. So there will be many different types of pollen grains, okay, will come and settle on the stigma. So the plant should have the ability to identify, okay, out of these various pollen grains from various sources which settle on the stigma. So it should have that ability to identify what is the right type of pollen, okay. So if it is a right type of pollen, then I say it is compatible. So then I say it is compatible. If it is a right type of pollen, then I say it is compatible, right. And if it is what I say, the wrong type of pollen, right. So if it is wrong type of pollen, then I say it is incompatible. So if it is a right type of pollen, I use the word compatible, and if it is a wrong type of pollen, I say it is incompatible. So what is actually the right type of pollen? So let's see the right type of pollen here, because we need to consider the cross pollination, right. So here what I say is that the pollen from another plant of same species, in that condition you have to take you have to say it as the right type of pollen, right. Then what is the wrong type of pollen? So here in case of this wrong type of pollen, so this is the pollen from same flower or another flower, okay, on same plant. So then I will consider that as a wrong type of pollen, right. So of course on a stigma, the pollen from the same flower, the pollen from another flower on the same plant, and the pollen from, okay, another plant, of course belonging to the same species or maybe of different species, will be settling on this stigma. So here, as I told, with the various colors I indicated that they are of pollen from various sources. Now, right, the plant should have this ability to identify which is the right type of pollen, and if it is a right type of pollen, then it will allow the pollen germination and the growth of the pollen tube, right. And if it is a wrong type of pollen, then the plant will reject that pollen. Okay, so the pistil, so so this is the ovary, and of course this whole is pistil. So here what I say is that the pistil has ability to identify the right type of pollen. So pistil has ability to identify the right type of pollen, right, and reject the wrong type. So here, let's say out of these various pollen grains that settle on this, okay, say this is a pollen from same flower. So this is a pollen from the same flower, right? And say this is a pollen from, okay, another flower but same species, right? And say this is the pollen from another flower, okay, of different species, okay, and you can say that this is pollen from another flower, okay, present on same plant, right? So now I think you understood that which of these pollen will be accepted by this pistil. So if you see here, the blue colored one, so the pollen from another flower but belonging to the same species is that pollen which will be accepted by this, okay, pistil. So this is the one which I call it the right type of pollen. So this is the right type of pollen, and remaining all are the wrong type of pollen. Right, so how this happens is because the chemical components of the pollen will be interacting with that of the pistil. So that will help the pistil to identify whether the pollen is of the right type or wrong type. So now if the pollen is of the right type, then what happens? So now see, in this case, once this pollen is of is identified as the right type, then the pollen germination takes place. So the pollen germination takes place on the stigma. So which means that through one of the germ pore, the pollen tube comes out. So here this is the pollen grain, and through one of the germ pore, the vegetative cell results in the formation of the pollen tube, right? And you can see this is the pollen tube, and that pollen tube is going to carry the male gamete. So it will be having the male gamete. So here these are the male gametes, right? So here the emergence of that pollen tube from the pollen grain is what we call the pollen germination. So once that is completed, then the pollen tube starts growing, okay, through the tissue of, okay, pistil. So this is the pollen grain, okay. Of course a little detail I will be discussing in the coming videos, right? So this one grows through this, okay, and here you see inside the ovary, so there is say ovule present, okay. So this grows, and then finally through the micropylar end, right, the pollen tube will finally enter into the ovule. So we know that inside the ovary is the presence of one or more ovules. So in this case, for an easy understanding, I have taken only one ovule. So you can see that, right? So this is the pollen tube that has grown through the, what I say, the style. So that has gone through the style and then entered into the ovary and finally entered into the ovule through the micropyle, okay, and into the embryo sac. Finally it enters through one of the synergids, and after which, so it will in fact break open and then releases the male gametes into the embryo sac. So that part I'll be detailedly teaching you in the process of fertilization, right? So this is what happens once the pollen is accepted. So the right type of pollen is accepted, and if it is the case, then the pollen grain will germinate and the pollen tube will grow through the style and enters the ovary and finally enters into the ovule, and ultimately it will reach into the embryo sac through one of the synergid cell. Okay, so with this I will end today's video. So I will continue the remaining part of the pollen pistil interaction in the next video, and we'll also see about double fertilization in the next video. Thank you.